Vaccines are concentrated biological preparations that prime the immune system to recognize and neutralize pathogens before they cause disease. The stakes are high. Smallpox, measles, polio, and influenza left a grim record of death and disability that shaped societies and altered demographics. Immunization offers a practical and powerful remedy: prevent illness, save millions of lives, and preserve economic and social stability. Early empirical practices were austere and inventive. Variolation appears in China by the 10th century and later spread to the Ottoman world and Europe. In 1796, Edward Jenner observed that milkmaids exposed to cowpox were protected from smallpox, and he deliberately used cowpox material to immunize a boy. Those humble beginnings marked the shift from artisanal craft to scientific enterprise. The 19th and 20th centuries added methods: attenuation, inactivation, toxoids, and conjugates, each improving safety and effectiveness. By the late 20th century, recombinant subunit vaccines and viral vectors arrived. The 21st century brought a major leap: messenger RNA platforms that encode antigens and instruct cells directly. Along this historical arc, there are 5 core themes that will drive our narrative. Mechanism: how antigen presentation, neutralizing antibodies, T cells, and innate adjuvants create durable protection. Timelines: why classical vaccine development often spans a decade, and how emergencies can compress that to months. Manufacturing: the demands of good manufacturing practice, bioreactors, fill finish, cold chain logistics, and global equity. Hesitancy: the cultural memory, historical abuses, and modern misinformation that erode trust. And next-generation technologies: self-amplifying RNA, nanoparticle scaffolds, and the ongoing quest for universal vaccines. This compact groundwork provides the context and primes the viewer for a focused dissection of immunology, pivotal experiments, and the logistical crucible ahead. The staccato of scattered observations hardened into the steady pulse of scientific method when germ theory replaced the miasma idea and recast disease as microbial drama. Pasteur's meticulous experiments, the attenuation of fowl cholera through serial passage and the rabies vaccine produced from weakened nerve tissue, turned artisan conjecture into reproducible technique, showing that weakened pathogens could train the immune system without destroying the host. Koch's postulates made pathogen identification a disciplined process, and serology, microscopy, and the emerging field of bacteriology supplied tools that converted anecdote into evidence. From that crucible came immunology. Cells, humoral factors, and organs became actors in a mechanistic story of antigen presentation, opsonization, and memory, and laboratories began to decode not only which microbes cause disease, but how the body remembers them. These intellectual breakthroughs led quickly to real, tangible victories. Smallpox, once a merciless scourge, became the target of organized assault. Jenner's idea of vaccination, refined into systematic campaigns and ring vaccination paired with surveillance and coordinated globally by the World Health Organization, culminated in eradication, an epical triumph that proved eradication was possible and set a precedent for global public health. Pasteur's rabies vaccine offered another lesson: an effective post-exposure prophylaxis that changed emergency medicine and showed vaccines could be both preventive and therapeutic. The mid-20th century polio campaigns crystallized the modern architecture of mass immunization: national registries, school-based programs, mobile teams, and cold chain logistics. And the Salk and Sabin vaccines demonstrated complementary approaches: inactivated injections and live attenuated oral immunizations, each with different operational and immunological trade-offs. Those victories did more than blunt pathogens. They created new institutions: ministries of health, vaccine advisory committees, regulatory pathways, and production facilities became fixtures of government, while philanthropic and multilateral organizations learned to finance and coordinate at scale. The public came to expect steady, measurable declines in childhood mortality, and policymakers accepted that vaccines could be planned, produced, and deployed as predictable public goods. That confidence sets the stage for the next set of questions: how protection actually plays out at the cellular level, how to compress timelines in emergencies, and what technological advances will be required to meet new challenges. Mechanistic clarity starts with the immune system's 2 theaters: the brisk, broad, innate response and the refined, memory-rich adaptive arm. Innate cells, macrophages, dendritic sentinels, and neutrophils strike first, secreting cytokines, phagocytosing invaders, and ferrying antigenic fragments to lymphoid organs where the adaptive actors prepare. Dendritic presentation on MHC class 1 and 2 is the pivotal cue that activates naive CD8 cytotoxic cells and CD4 helper cells, and that licenses B cells to undergo affinity maturation. In germinal centers, B cells go through somatic hypermutation and class switching under T follicular helper guidance, sculpting high affinity, neutralizing antibodies that block receptor binding or prevent membrane fusion. Memory B and T cells form a durable ledger of protection, poised for rapid clonal expansion on re-encounter. Tissue-resident memory adds a local, ready defense at frontline sites. Adjuvants act as magnifiers, engaging pattern recognition receptors to amplify antigen presentation, skew helper subsets, and shorten the time to protective titers. These mechanistic primitives map onto platform-specific trade-offs with clear operational consequences. Live attenuated vaccines recreate the infection narrative and yield broad humoral and cellular repertoires, but risk reversion and require careful cold chain stewardship. Inactivated vaccines are safer and more stable, but usually demand adjuvants and multiple doses to build durable memory. Recombinant subunit vaccines permit precise targeting of conformational epitopes and controlled glycosylation, but rely on potent adjuvants and nanoparticle scaffolds to present antigens effectively. Viral vectors deliver genes into host cells to elicit robust CD8 responses, yet can be blunted by pre-existing anti-vector immunity and complicate manufacturing. mRNA platforms enable fast design and strong antibody and T-cell induction, but hinge on lipid nanoparticle chemistry, supply of modified nucleotides, and stringent cold storage. Real-world constraints further bend design choices. Antigenic drift and variant escape force iterative strain selection. Imperfect correlates of protection make regulatory endpoints and bridging studies contentious. Protracted R&D, phased trials, and lot release testing collide with emergency timetables. Manufacturing scale-up confronts bioreactor yields, process validation, aseptic fill finish, and global fill capacity limits. Cold chain logistics, thermostability, and equitable allocation determine who actually benefits. Those mechanistic and operational limits compress the problem space and set the agenda for the next act, where timelines, manufacturing crucibles, and social trust must be reconciled with immunological ideals. Act Two's mechanistic and operational tensions erupted into a trial by fire during the COVID-19 pandemic, when genomic sequencing, mRNA design, and global urgency collided in a tense, high-stakes race. Within days of a published sequence, labs were already building spike protein constructs and testing lipid nanoparticle formulas. That luminous sprint showed how design speed could be a transformative advantage and also a new Achilles heel. Regulators responded with rolling reviews and emergency authorizations, compressing years from classical timelines. Phased trials ran in parallel and large, multinational efficacy endpoints were used to infer protection faster than ever. The early wins were striking: high efficacy signals, rapid authorizations, and immediate deployment. But the speed also revealed hidden weaknesses. Small signal safety events only became visible after millions had been dosed, forcing robust post-market surveillance and real-time pharmacovigilance. Manufacturing turned into a baronial struggle. Bioreactor yields, proprietary ionizable lipids, single-use consumables, sterile vials and stoppers tied scale-up to fragile global supply lines, and fill-finish capacity repeatedly became a choke point that turned doses into precious coins. Cold chain needs for early mRNA vaccines imposed an onerous burden. Ultra-cold freezers, validated transport shippers, and trained handlers made distribution a logistical opera that many low-resource settings could not join. The result was stark inequity. High-income states negotiated advance purchase agreements, built stockpiles, and rolled out boosters, while many low- and middle-income countries waited months or years for scarce supplies. That geopolitical fraying undermined the moral authority of global public health. Public trust, once bolstered by past vaccine successes, eroded under perceptions of haste and a stream of amplified misinformation. Rare adverse events like vaccine-induced thrombosis and myocarditis were seized on as emblematic failures despite favorable risk-benefit balances. The Crucible forced a hard reappraisal. How much speed is acceptable, and what surveillance, manufacturing decentralization, and transparency must accompany compressed timelines? Questions about equitable allocation, technology transfer, thermostable formulations, and investment in regional fill-finish capacity moved from academic debate to urgent policy priorities. The pandemic left the system at a crossroads, a rough but velvet-lined rupture demanding structural reform, clearer public stewardship, and scientific humility before we face reform or repeat catastrophe. The turning point came when platform vaccines, especially mRNA, proved they could go from sequence to dose in record time while delivering strong protection. That proof validated a new model for fast, adaptable vaccine development and set off immediate, concrete changes. Funding shifted toward modular platforms and next-generation antigen design, and major investments flowed into regional manufacturing hubs that prioritize portable GMP suites, fill-finish lines, and reagent stockpiles to prevent future bottlenecks. Regulators changed how they work. Adaptive trial designs and immuno-bridging endpoints began to replace only long sequential phase gates, and rolling data reviews became an accepted, rigorous alternative to waiting for a single final submission. Surveillance became an integrated network. Linked genomic sequencing, near real-time adverse event reporting, and federated electronic health records that triangulate safety signals and variant emergence faster than before. In practice, technology transfer agreements and standardized LNP chemistries spread capability beyond a few capitals. Thermostable formulations and alternative delivery devices eased cold chain constraints, and coordinated procurement mechanisms plus patent pools helped improve global access. But the rapid success had a downside. It inflamed political polarization and amplified hesitancy as opponents seized on uncertainty and rare adverse events to cast long-lasting doubt. To fight that wave of mistrust, communication efforts had to be precise and community-focused. Transparent data portals, local vaccine ambassadors, independent safety audits, and clear, targeted risk-benefit messaging aimed at rebuilding trust. The result is a new operational playbook that delivers speed and flexibility, but also exposes social frictions that the response must confront. Having turned the Crucible's lessons into a practical playbook, the next chapter promises concrete, gilded advances, thermostable vaccines, and mucosal formulations that free distribution from the tyranny of cold chains. Universal and pan-pathogen vaccines that compress strain-by-strain politics into strategic durability. Self-amplifying and multivalent messenger RNA constructs scaffolded by nanoparticle and structure-based antigen design that raise potency while shrinking dose volumes. AI-driven antigen selection that can accelerate candidate choice from months to days. And decentralized portable production units that democratize manufacture and blunt geopolitical choke points. Together, these innovations could shorten development timelines, simplify logistics, and turn episodic emergency responses into predictable public goods. But technology alone will not be enough. We need a bold rethinking of policy, sustained global financing, enforceable rules to ensure manufacturing equity, clear legal frameworks, and authentic community-centered engagement if uptake and trust are to follow. The lasting effect will be judged not by lab triumphs, but by whether nations stitch these tools into durable, equitable systems. Will we seize this gilded opportunity to make vaccines a steady, shared inheritance for everyone today?